Hello, welcome back. If you have not subscribed my channel, please click on the subscribe button now. So today we will study the bacterial growth curve and various methods of bacterial cell count. When we place the bacteria in suitable environment, first thing they do is shed dormancy and become metabolically active. We can see two types of typical bacterial growth patterns. In the first, the bacterial cell synthesizes new cell components and increases its size. In the second, the number of bacterial cell increases multiple times. The basis of bacterial population growth is binary fission. Binary means that one cell becomes two. Parent cell enlarges, duplicates its chromosome, and then starts to pull its cell envelope together in the center of the cell using a band of protein. Eventually, it forms a complete central septum, thus dividing into two daughter cells. With each successive round of division, the population increases. Time taken by the bacteria to complete binary fission is called generation time. Once the bacteria start dividing in suitable environment, the number of bacteria doubles with each generation time. Generation time is also called doubling time in simple words. One bacteria gives rise to two bacteria by binary fission. The time taken for the process is called generation time. After 2 generation times, 4 bacteria are produced, and after 3 generation times, there are 8 bacteria, all produced from a single parent bacterium. Now let us see how many bacteria are produced after each generation time is over from a single bacterium. We can take the example of E. coli, that is a generation time of 18 minutes. 10 generations will be equal to 180 minutes or 3 hours. After 10 generation times, the single bacterium of E. coli will produce 1024 bacteria. Now if we assume perfect conditions for continued bacterial multiplication, how many bacteria will be produced after 24 hours of incubation? We can see that the number increases exponentially, and after 24 hours or 70 generations, there should be uncountable number of bacteria. This would be an unimaginable number and mass of bacteria. But there is a limitation to the process of doubling. We can plot the data arithmetically, and that gives us a constantly curved slope called the population growth curve. In reality, a population of bacteria does not double endlessly. Numerous factors prevent the cells from continuously dividing at their maximum rate. A population typically displays a predictable pattern or growth curve over time. The first phase of bacterial growth is slow and hardly noticeable. It is followed by the phase where bacteria actively divide and keep doubling their numbers over successive generations. This rapid growth leads to exhaustion of nutrients in the culture medium, and the bacterial numbers become stagnant. With continued incubation, the bacterial number decreases rapidly because of accumulation of the bacterial cell waste in the environment, causing cell death. The first phase of bacterial growth curve is called the lag phase. It is followed by the log phase or the exponential growth phase. The third phase is stationary phase, and the fourth phase is called the death phase. Let us see what happens to the bacteria when we inoculate a nutrient broth with a loop full of bacteria and keep it in an incubator. The bacteria recover the damage, start synthesizing RNA, enzymes, and other molecules needed for their growth and increase in size. This is called the lag phase, where they shed their dormancy and become active. Now they are ready to multiply and enter the next phase of growth. This is called the exponential or the log phase of growth. Their population doubles with the passage of each generation time. Once the conditions become unsuitable for continued bacterial growth because of exhaustion of one of the essential nutrients, the exponential growth stops and the stationary phase begins. The bacteria, however, continue to produce metabolic waste that makes their environment toxic and they drown in their own waste. In the stationary phase, some bacteria start dying. The number of bacteria that are born and that die remain equal, and that keeps their number stationary for some time. If we continue the incubation, more bacteria will start dying soon. That is the beginning of the death phase. In the death phase, bacterial number decreases exponentially. At the end of the death phase, there are very small number of bacteria that will remain alive for many weeks to months. These bacteria enter a dormant stage and keep waiting for the proper environmental conditions to restart their growth cycle. In the laboratory, refrigeration is used to slow the progression of the death phase so that the cultures remain viable as long as possible. So these are the various phases of bacterial growth curve in the artificial culture media. Now we will study the methods used for counting the bacteria during each phase of the growth curve. The method traditionally used was the viable count technique, in which the total number of live cells is counted in each phase at successive time intervals. In brief, this method is also called the batch culture, which includes the following steps. First, we place a tiny number of cells into a culture medium and then incubate this culture over a period of several hours. Next, we sample the broth at regular intervals during incubation. The sample is then plated onto the solid medium. The colonies are counted after incubation to get the estimate of live bacterial cell count. Each colony on the plate represents one cell or colony forming unit from the original sample. So what is the practical importance of the growth curve? For those who are working on bacterial cultures, they should not incubate the culture beyond the stationary phase of the bacteria. Otherwise, the cells will die in large numbers. Secondly, we use samples from the exponential phase to demonstrate the size, morphology, motility, and also for staining purpose. The older cultures, however, are used to demonstrate the formation of spores in sporulating bacteria. This also has implication in the spread of infections. For example, a person in the early and middle stage of an infection is more likely to spread it to others than in the person in the late phase of the bacterial infection. This example of the growth curve is from a closed batch culture, where we are neither adding fresh nutrients nor removing any bacteria out of the culture. But in industrial processes, we can use what is called a continuous culture method. A chemostat is an example of such a bioreactor to which fresh medium is continuously added, while culture medium containing leftover nutrients, metabolic end products and microorganisms is continuously removed at the same rate to keep the culture volume constant. We can achieve a desired growth rate of bacteria by varying the inflow of fresh sterile medium, inflow of the sterile air, temperature settings, and so on. The continuous culture media are well mixed to keep environmental conditions constant. Chemostats are widely used in the industry for the production of ethanol. Enumeration of bacteria. Two types of counts can be made. The total count gives the total number of cells in the sample irrespective of whether they are living or not, and the viable count measures the number of living cells. Direct counting under the microscope using various counting chambers can be used to get the total number of bacterial count. We can also count using stained smears after spreading a known volume of cultures over the measured area of slide. We can use nephelometer to count bacteria that measures the concentration of suspended particulates in liquid or gas colloid. by measuring the wet or the dry weight of the cells after separating them through filtration or centrifugation, by chemical assay of the components like nitrogen assay. Counting can be automated by sensitive devices such as Holter counter, which electronically scans a culture as it passes through a tiny pipette. As each cell flows by, it is detected and registered on an electronic sensor. A flow cytometer works on a similar principle, but in addition to counting, it can measure cell size and even differentiate between live and dead cells. When used in conjunction with fluorescent dyes and antibodies to tag cells, it has been used to differentiate between gram-positive and gram-negative bacteria. We can estimate the viable count in bacterial suspension using the dilution method. It uses the principle of successive dilution and plating from each dilution to reach a dilution where cells can be counted easily. This method is used routinely for presumptive coliform count in water bacteriology. We can also use the plating methods for viable count. First method used is Miles and Mizra method, where we use an automated pipette that delivers only 20 microliters of fluid as a drop onto the medium. We make serial dilutions, and using the automated pipette, we inoculate the plate divided into segments with one drop from each dilution. The solution is dropped from a height of 2.5 centimeters and allowed to spread automatically on the plate. After incubation, we get the results that gives us the viable count as shown in the picture. Other plating method for viable cell count are the pour plate method and the spread plate method. In the pour plate method, we pipette the inoculum into the sterile plate and add sterile medium into the plate. The plate is swirled and mixed and then incubated. The bacterial colonies grow in and on the medium. In the spread plate method, we pipette inoculum onto the surface of the agar plate. It is then spread evenly over the agar surface and incubated. The colonies grow on the surface and are counted. So that was all about bacterial growth. Hope you liked the video. Please subscribe, like, comment and share. Thank you for watching.